So I think this is good opportunity to actually also talk a little bit about Ascendia. I know many of you may not know what we do, and also take a deep dive into the technology that we offer, and also can help actually people actually struggling with the poorly soluble molecules. So I'm going to talk today some few slides showing the Ascendia's core capabilities, and also give some case case studies. Which really we cannot talk too much because all the projects we have is customer projects, and it's not really, so we don't have our own actually data. We have actually the data that so we cannot really share everything that we so we do a lot actually more on the, I was talking to some of you earlier, we have technology that can be used both actually for oral also in parenteral area as well. So let's move on. Okay, so my talk actually today is going to be very brief. I would like to just give very highlights, not to take a very too much in the deep dive and look into the core technology, but look at the how it really can help people actually struggling with the poorly soluble drugs. So here I'm going to talk, just give a brief introduction about our Ascendia, and then what are the challenges with poorly soluble molecules. I think we all here for the same reason. We understand the drug discovery, a lot of molecules coming out of discovery, they're poorly soluble. In my tenure actually at BASF, this is actually my core in expertise, working with all the companies actually, all the pharma industry, promoting polymers and the the surfactant and the lipids. They all go into the amorphous dispersions and also in the sets, SMEDs, nanoparticles and so on. And also working with the liposome company not too long ago, not too far actually from here, back in 90s when I came to this area, there was a company, liposome company right here across the street. I worked for 7 years, so I've been really working in the lipids area for a long time, including my graduate studies, those days in PhDs as well and postdocs. So with that said, I'm going to look into just giving introduction about the solubilization, the challenges, looking at the how Ascendia can help, and also about the technologies as well, what we apply, and give some case studies, and then take home message. So Ascendia is a small company. It started about 10 plus years ago. Located actually right on Route 1. If you're driving Route north from here, about 15-20 minutes, we'll see building a called New Jersey Biocenter, Bioscience Center, which is really ideal, because in the northeast it's easily located. It's easily, I would say, you can drive through, or you can take Newark. You can come to Newark, or Philadelphia is highly it's located right in the center of the central Jersey, and we have actually expanding for the last 2 to 3 years. Since I joined in September this company, we have really hired many people. At least 10 of them, in the last 7-8 months. And we expect that we, in the next 5 years, will be at least reach 200 people in the company. This is the lab that we have, both and the R&D labs and also the manufacturing as well, which not many CDMOs may have of this size, but our emphasis is actually not only use just develop the product in the R&D, but also take to the phase 1 and phase 2, and also going to commercialization as well. So we are building a 10,000 square feet facility where we can manufacture the phase 3 and also commercialize the drug products. We do have the employees, actually most of them, I would say 50% are more in the core science. Most of them, they have PhDs. So we have very big formulation group. Also we have a very big analytical team, and also the manufacturing team as well. So our core strength includes the GMP manufacturing, which both includes the sterile and non-sterile. We are expanding actually the sterile capabilities, because a lot of industries actually looking into parenteral and also long-acting injectables, and they are not many companies actually they can offer the services, because maintaining the facilities is very, very expensive. So we have the clean rooms, ISO in the qualified ISO 5, 6, 7, and 8 as well. And also we have the technology, because we not only can just formulate, but also we use our own technology. It's a pay for service. So basically, so you don't really charge any milestone payment from the customer. We just ask them, if they really want to use our technology, we can really use and formulate. So it's like it took a very holistic approach to use our capabilities and give the formulation optimize, and then give the formulation to customers. They are going to the IND. Okay, so our 90%. The molecules that we work on, they are new chemical entities, maybe about 10% or maybe 20% or less, they are complex generic. So maybe they have been approved, but there are people are looking for this different modalities or different applications and so forth. And again, our technology, I'm going to talk more on the lipids and lipid nanoparticles and also polymeric, PLGA for example. And some of you spoke yesterday and also probably today, how the PLGA can be used for the making the microspheres or nanospheres. We also publish papers, so if you have get chance, we can also go. We have some publications outside. One published in American Pharmaceutical Review is a very comprehensive review on the PLGA and nanospheres. So coming back to our capabilities, we can see I don't have to read every line, but we can do most of the things that customers are looking because. When they come to us, they're not looking for just for the R&D work, because anyone can do R&D, but how to take it on scale up, that's challenge, right? And we spoke yesterday as well, because R&D is one step, but then you're making it in large scale. Not many things actually can be very consistent or very robust, and so we have to really master the technology, and with that technology, we can also take it to the next level and bring the products to the to the market faster. So, that said, we can handle in the sterile injectable, long acting, and also we have non-sterile oral formulations. We can manufacture on all those, and we can do the aseptic filling, which is also key. Not many other places may have it, and we can do it in pre-filled syringes and also the vials as well, and lyophilization. So we can do in one shot actually. We can everything in our side. So coming back to the challenges, probably have seen this slide many times. I know Firoz spoke about it's class 2 and class 4 drugs, particular class 2 drugs, which are poorly soluble but also permeable. And again, one is a dissolution limited, and the other one is solubility limited. And again, those technologies that really is applicable, particularly the one you're talking about, nanotechnology, morphodispersions. The co-precipitation, precipitation that Dr. Prudhomme spoke. So these are all technology applicable for those molecules. They are not easily handled, right? Cannot be really developed just by simple micronizations, simple complexing with cyclodextrins, right? So with that said, so we are looking, for the challenges because and how to find the solutions for those molecules. They are poorly soluble. And also even the class 4 drugs are even more challenging as well. So these are the core technology that we are talking about. We have the patent on these technology, each of them, and the last one actually just we got on lipid sol, which I'll come back a bit later. But MRSol, you can see from the name, it sounds amorphous dispersions, nanosol, nano suspension, nano emulsions are also the emulsol. The microemulsions are emulsions as well. So what are those and how can be applied for a molecule that you want to develop, right? So MRSolve again, we have the patented technology where we can look into the molecule and see what are the food effects, particularly how we can really design. And again, all the excipients that we use in our technology, they all been approved in the IAG database, FDS. So we're not using any novel excipients. So there's no concern, right? I mean, having worked in my previous company, because we had Soluplus, we had a lot of other polychloride smart seal, and other novel polymers. They bring some challenges, to the industry, and people are really keep a distance not to use it because then it brings a lot of regulatory hurdles, right? Though the FDA is changing now, they are looking, they have novel excipients lately, but it's a long way to go. So MRSolve is amorphous dispersions, and we choose the polymers accordingly, and again make sure the polymers are safe and also have no food effect as well. because that's really the key, to make sure that it maintains the supersaturation without compromising the efficacy or of the drug. And then the other one, the Emulsol and I think slide is a little bit off, Nanosol and also the Emulsol. And again, it can be applied to both the oral and also the injectable, even the parenteral, even for the topical as well. So there's a lot of interest in the topical, microstructures. If you consider microstructure in the space, all these lipids actually they play a key role, right? How you can really pack the molecule in the layers of all the lipid structures, and how they influence the drug permeation through the skin. So these are all all lipids, right? So they really control through the microstructures, and so we can all this technology we do have some projects on topical as well. But a lot of actually our customers, they are using the innovative molecule, and then also looking into more in the the parenteral and long injectable, and also nanosuspensions for oral applications. So lipid sol actually comes very recently. We are looking into different. We do have the access to the the special we call the novel in excipient, but again these are the just new. But we can design any kind of the lipid structures using the technology, and particular not only designing but also scale up. So we do have the capabilities to the precision nanosystem. We can go from ignite very small scale to all the way to hundreds of liters in manufacturing as well. So we do have capabilities where and also the expertise we are in sense. So if you look at this all this in this structure, they all combine together. And all the speakers today and also yesterday, they talk about the PEG lipids and regular lipids, cholesterol. All these are very important component of making the membranes, right? Sometimes the membrane structure is not enough. I worked in the liposome for a long time, and lipids area. The liposomes are not a really good vehicle to really stabilize the drug. Particularly when you're loading. When it comes to loading, I worked with paclitaxel many years ago when I worked in liposome. It was very difficult to encapsulate taxol like structure. Molecular weights 853 Dalton. We can put only in liposome 5%, right? 5% is not enough because you're using 95% lipids, and that could also be very toxic, right? So these all things actually matters, particular not only the not in so much in the R&D, but when you start scaling up, how you can really have a robust process, thereby you can stabilize the drug and also be able to circulate long enough. People talk about pegylated lipids. It was discovered by Terry Allen back in many years ago. Peter Cullis and all those people, they are the pioneer developing this lipids, just to show that it can be called the stealth liposome. So again, the same technology people are using vaccines, because they all pegylated, and the reason being because they can circulate longer in the systemic circulation. So we can design any structures, and again the composition of the lipid will be different for each one. We published a few papers, so we can take into look. We have the this issue on our desk outside. So now the question is how we can design, and how we can decide also which technology works for it. So we look at a very holistic approach. Just to make sure that we are not really returning each and every bricks, because we want to make sure that we find the right solution, because we are the solution provider, and the customer actually they are coming to us hoping that they can take it to the phase 1 as quickly as possible, right, without losing. So our aim is to find the solutions within 3 months, screen the molecule, and then come up with the right technology where they can be scale up. And also we're able to part of that scale up process, going to phase 1 and phase 2, and eventually are going to phase 3 as well, also hopefully by end of next year. So depending, and this probably you have seen this chart many times, and everything depends on the molecular weight and the log P, and also the melting point. Higher the melting point, talking about the big dust structures, they're very very strong lattice, the lattice energy. To break the lattice energy is tough, because I think Firoz he spoke yesterday about sugar and other examples, right, and sodium chloride. It's very similar, I'd say paradigms, where you can dissolve the sugar in water, but sometimes if you can dissolve a molecule in water solvent, it may not stay in there, because when it hits water, it will crash out, right? So finding the right technology is very relevant, because then solubility limited, right? So we can choose, depending on the pK values and all those, since we have everything on display, and very flexible, very agile as well. So we can choose whether it's nano suspensions or nano emulsions or micro emulsions, or the or MR-sol, depending on the. One thing I would like to also mention, that a lot of our customers actually, they are looking more into the liquid formulations, right? So whether they're going into injectable or long-acting suspension. So we are looking into more the milling, process, because that's the way we can use both. I'll come back to that one, more like a top-down or bottom approach we can use, and it scale up very easily. Okay. I'll give an example. I think this also came up yesterday when AbbVie or Abbott was developing this HIV drug, and there are two drugs. Actually, one is class 2 and class 4 drugs, lopinavir and also ritonavir. Even ritonavir was developed as a Norvir tablet, right? Not too long ago, right? It was developed as a capsule, but here this is the fixed dose combination of both ritonavir and lopinavir, and they came up with the capsule back in the 80s. Still I was a graduate student. HIV came up. That in those years, people are really struggling to find a cure for HIV, right? So these capsules were developed, but again, it requires the refrigeration, and also if you're shipping to Saharan countries, sub-Saharan, then you really have to refrigerate to certain temperature. Whereas a tablet, you have just shipped around, and also it reduces the the pill burden on the patients, and also they reduce the the food effect, right? Whereas in capsules, there is a lot of food variability. There are some data I don't have here, but they can see how the capsule has food variability depending on the what you eat, right? In the meal also dictates the food absorption in the drug absorption as well. So again, in those depending if you go back, I mean, if you look at the so just to nutshell, I mean, so the customer, they are open to, right? They want to go in phase 1 so they can capitalize, and also on the investment as well, right? So looking at the nanoparticle technology, I will not really go into all these details. I've been working many years in this area. Lipids have come back now, right? Back in the 80s when I started my graduate study with lipid synthesis, I was synthesizing the lipids, how to do the total synthesis of phospholipids, right? That was my project. At Queen's College at City University. So with that said, I mean, it is not easy to synthesize these molecules. If you are, synthetic chemists and working with the, phospholipids, it's so difficult to make. It takes several, steps, and the yields are really poor. But technology has improved, and also so when you're making a formulation, make sure that you're not wasting the lipids, because they are extremely expensive, and also they're stable, of course. At the same time, drug loading is always a challenge, right? So you're looking two things: stability, and also the enhancing the drug loading. Thereby, you can maximize your efficacy, because you don't want to overdose the lipids, and underdose the drug, right? So that's really the critical attributes you have looking into any products. So these are the structures we probably have all know about this structure. Among them, actually, I would say cubosomes are, there's no products yet, but there's a lot of literature. So I'm working on writing a review article actually on cubosome, hopefully will publish soon. And so we can design all this. But if you look at the over the years, I mean, there are a lot of things actually have changed. But I was really struck actually looking at the timing, right? So liposomes were discovered in 60s by Bangham. And then came many other things afterward. But we look at the SLN and the NLC. These are the really people are talking about nowadays, because number one, they are very stable. That's monolayer structures. They're not bilayers, because bilayers always create problems, because the drug start leaking, and then you cannot really ship the liposome unless you freeze it. But the solid lipid nanoparticles and also the lipid nanostructured lipid capsules, they are better, I would say ideally better suited because of the structure, and because of the monolithic structure. And then you can create actually, by depending on the structure of the lipids, we can also create these structures and nano-assemblies where you can increase the drug loading. But also it brings some caveats, because if you have the SLN versus the NLC, NLC is slightly better actually, because particle size might be the same, but only the composition of the lipid by changing, you might be able to generate, create one versus the other. But if here looking at the bottom one, particular, because the inside is so flexible, because we are using something oily phase, so the stability might be different. For the NLC might be much better actually than the SLN. Because of the solid structure, because when the drug is encapsulated inside the structured matrix, it may expel out when you do the stability, for example, because as you know, in the tableting and others, we need to have enough room, because otherwise if the tablet starts contracting, then the coating layer can shrink and also can rupture, right? So this is a very similar the paradigms, because this since is a monolayer, like putting a coating layer around the tablet. And if something is very solid and expands, it's going to expel out the drug, and therefore you lose the stability of the molecule, right, altogether of the nanoparticles. So therefore, actually, the leakage is further minimized by using the NLC approach than the SLN. But of course, both are applicable for the designing. This is actually slide was shown yesterday, but I brought in actually just to bring one point here, because we are working with a company. We have licensed about 40 plus lipids, the non-ionizable. Why non-ionizable is so important? Because if you look at the structure of these vaccines, right, they all have the non-ionizable lipids, right? And it's very interesting, because back in the days when I worked in liposome in 80s, 90s, early 90s, we worked with the non-ionic lipids. They're also helping to do the transfections. And it was very hard to do the transfection, because the key is actually why we need the ionizable lipid, because to interject the transfection from one cell to another. And we had to create, so I was, as an organic chemist, trying to design non-ionic actually lipids, which is also very novel, and then able to transfer the DNA from one cell to another, from a host to host chemistry approach. So these are, so we now have the access to non-ionizable lipid through a company. So hopefully, so if you have any projects, anything, we will be also able to do the this kind of also the lipid nanoparticles. Again, I don't want to really go through, but basically once it's endocytosed, then it releases the mRNA, then it gives the spike the proteins, and then it binds to antibodies. So this is I just copied actually from this paper. This paper is really, I like this paper. If some of you have not read, but I would encourage you to take a look actually. This is a very nice paper. So Ascendia, we do have the, I'll quickly run through, because we do have the, both the bottom approach and the top approach, and both are actually okay, because I think, depending, how robust the process is, we can both, take it. But, when you're doing a small scale, the drugs are very precious, right? So they're very, so we don't want to waste the drug, because, customer is bringing like few grams, and you want to make sure that you create something and give to give back to the customers, so they happy. So we do have all this approach. We have so much equipment we can do, like a scale up, small is to large scale, but and we can do very easily with. So if you're using a bottom approach, we can make a film forming, right? So, and that's typically used. You can make the PLGA, use all the. Or if you're using the microfluid approach, the top approach, we can do as well. We can do the high pressure homogenization and everything. So, and that way you can screen the whole bunch of the actives in a very short time. Okay. So just giving you some example. We have so many examples, but I don't know, because again these are the proprietary and also in the customer projects. But I'll just walk through, give very highlight what we have. So we have several examples, and this one example of PLGA I mentioned earlier. We have very good hands on the PLGA, and we have several projects actually in long acting. And the customer here in this case, looking for is a polypeptide, and it's a class 4 drug, and they're trying to reduce the Cmax. And just the reason for Cmax reducing, because it was very cytotoxic, so they wanted to make sure that that meets the therapeutic windows, right? And also the is increase the half life as well. and you are able to formulate with this one, and able to deliver at the right time. So so this is really like you can see, this is the picture we see at the bottom. This is the from nanosystem, precision nanosystem is a blaze. So we have this equipment as well in the lab. So at the end, we can see able to reduce the toxicity of the drug and increase the drug exposure for the PK during the PK study. So it's really I would say this kind of data we have, but we cannot really present actual data what happened, right? So again, this is the PLGA nanoparticles. So we can people use PLGA because yesterday the speaker spoke. That's one of the commonly used polymer. And safety-wise, I mean, there's no question. We can use up to 500 some milligrams. If you look at the IIT database, also one of the publications we have here, it gives a number as well. I think you can go up to 500 plus milligram per dose, right? Maximum potency. And there are many ways we can do the PLGA nanospheres. The hot melt extrusion is not for the nanospheres, but we can I don't know whether you can spuronize it to make a nanosphere, but typically it's more for the implant, right? But otherwise, the most commonly method for the PLGA is the emulsions, double emulsions, and then the other one is the co-observation, which is a anti-solvent approach to make it hard surface. We use the silicone oil and others. So there are many ways we can do, and we do have the several projects on this one. And so if some of you are interested, we can talk more. Simon, how much time I have? Come on, okay. I think I'll walk through quickly actually. So again, you can see this is a drug release profile. We can the reason because you want to increase this the extended release so you go from 7 to 14 days versus the so the customer was trying to look into where you can extend the 2 weeks release or so. Again, this is another example of we can do the wet milling, and this is again another projects. That we're able to deliver that we can go from by nanomilling as well. This is one example of the carvedilol. Carvedilol is a very probably sold as Correct tablet, and for the cardiovascular, and able to formulate it and also deliver. We can see it can go up to 24-hour release. The PK profile. And we have the patent on this one, this technology up to 2036. This is another example of the so this is also very important example because a customer they had problem with the particular this molecule, and they wanted to reduce the pill burden, right? So we do have technology where we can apply and look at do the assessment, and we're using the nano suspensions. This is the oral pill. And able to reduce the pill burden, we can cut down from more than 10 to 3 capsules, right? So which is big relief. It's more patient centric as well, and also it was very effective, reduced the tumor size as well. So it was able to handle from both sides, the efficacy as well as also the the complexity in terms of delivering the molecule, right? This one is also very important. Clopidogrel. This is the example of Plavix, right? We just had the press release a few days ago. If you go on Ascendia's website, so this we're using our technology able to develop IV formulation because there is no IV formulation. So we just we are going through the another company which we are going through the clinical trial. So we just had a press release, and you can see the real challenge. I'll tell you what was the challenge. The drug was unstable. It isomerizes, right? So it goes from Si enantiomer to the R enantiomer, very quickly. But using our technology, lipid technology, lipid nanosol, even sol actually, you're able to control the conversion, right? And even the formulation is very, very stable in these formulations, and also increase the solubility using this same approach. You can see the solubility in the pH 7 is very low, and then using the oil phase and other lipids, we can increase the solubility much higher level, and therefore we can reduce the dose strength as well. So when you're coming to the selection of the what are the how you make the decision, we can just you can follow yes or no. If you're developing an oral formulation, go on the left side. If you're going for the injectable, you can go from the right side, and then you decide whether the is what kind of molecule is a biologics or the small molecule. If it's biologics, of course you have to use the lipid nanoparticles, or even if the not small molecules, we can apply both. If it's something RNA or DNA, we can use also the cationic lipid, ionosyl lipids as well. So this is very simple guidelines. I also published this one. It's also on the Ascendia's webpage as well. So we can handle either the BCS class 2 or class 4, and also you can see if something is more less permeable like class 3 drugs, it would also handle, because then you require some kind of special transporter as well. With that, I would like to end my presentation. Again, we do have the technology, and we have all the capabilities, as I said earlier. And one thing we believe in the best, right? BEST, and the brilliant technology, excellent services, superior quality, and trust, right? So these are the 4 actually tenets of Ascendia. So we like to make sure that we maintain those assets ourselves. With that, I would like to end here, and thanks for your attention. Okay. Any questions?
Thank you, Shaukat. Thank you.